think we'll get started with today's lecture. So we're going to be moving on this week to studying the issue of structure, primarily protein structure. And we're going to be looking at some of the computational tools we would use to work with protein structures, as well as some information about how you access, view, and work with the structures in general. So I do want to give a brief announcement that there's going to be a guest lecture on Wednesday. Joel McManus, who is the third lecturer in this class, will be giving Wednesday's lecture. And then he'll be back later in the course to teach a full one-week module in this section and another one in the second half of the class. So just be aware of that if you don't see me here on Wednesday. Aside from that, I think we can just jump into the material today. Are there any questions on anything from last week before we get to that? Okay, so before we can get to the real computational material, I do need to give a bit of background on the issue of structure and the biology and biotechnology related to protein structures. So we need to know what we're talking about when we discuss protein structures and what's interesting about them. And beyond that, we also need to know a bit about the biotechnology for finding protein structures. So as is often the case when you're doing data-driven analysis, it matters quite a bit where your data comes from. Different technologies have different characteristics, different trade-offs, and so this is something we need to know about. What are the strengths and weaknesses, or advantages and disadvantages of different ways of finding structures, and what are the options available? So that's basically a preview of what we'll be talking about today. And I guess the automated slide changer isn't working today, so we'll do it the old-fashioned way. So to start with, what we're going to be talking about is the protein structure hierarchy. This is something that's likely many of you have seen in previous classes, and this is often broken down into 4 levels of protein structure. We have the primary structure, which we've already seen. So the primary structure is simply a synonym for the amino acid sequence that we saw when we looked at sequences in the first couple of weeks. There is the secondary structure, which are recurring patterns of local structure that happen to be particularly favorable energetically in protein structures, and so will occur again and again. We will sometimes see an extra level after secondary called supersecondary, so I'll just refer briefly to that. Kind of recurring combinations of secondary structure elements that you see in many proteins. Tertiary structure is often used synonymously with simply the structure of the protein, and that refers to the full three-dimensional fold of a single polypeptide chain. So most often when someone talks about the structure of a protein, they're talking about the tertiary structure. But it is worth noting that often what's really functional in the cell is what's referred to as the quaternary structure, which is the structure of multiple polypeptide chains coming together to form some kind of complex. And I'll say more about that a bit later. But that at a high level is the structure hierarchy. And what I want to start with is talking about each of these levels in the hierarchy. Right, so as I mentioned, we have already seen primary structure, but just to go into a bit more detail about the chemistry behind it. You should all at some point have seen a peptide backbone. This is the backbone of the hypothetical protein sequence. We have an amino group at one end. We commonly treat the amino group as the start of the protein. And then we pass through the backbone, alpha carbon, carboxycarbon, to next amino acids, amino group, alpha carbon, carboxycarbon, and so forth, till we get to the carboxyl end of the protein, which we would generally treat as the right end of the protein when we write in a canonical sequence. And hanging off of each of these alpha carbons, we will have a side chain, and you should all be familiar by now with the fact that there are 20 standard amino acids, each distinguished by its own side chain, and commonly when we refer to the primary structure of the sequence, what we're really referring to are these residues, basically the sequence of side chains for these proteins. We've all seen by now the 20-letter alphabet for this. This is most commonly how you'll see these written in databases. There is a 3-letter alphabet that's a little easier to read, but it's worth knowing the 20-letter alphabet, and just knowing that M is methionine, H is histidine, C is cysteine, and so forth, and just memorize those 26 questions about this? So the next level then would be the secondary structure. And the secondary structure really refers to positioning of the backbone of the protein independent of the side chains. And this is generally captured in 2 degrees of freedom per link in the chain, so per residue in our amino acid sequence. And what we essentially have are 2 rotational degrees of freedom on the backbone per residue that are conventionally referred to as the phi angle and the psi angle for that residue. The third link is essentially rigid, so you get a double bond, partial double bond here, so you get some resonance there that prevents that third link from rotating, but basically you get 2 degrees of rotation, and that describes the possible positions of the backbone of the protein. So you can approximately describe the shape of the backbone in terms of a sequence of phi and psi angles. And what leads to secondary structure is in part that only certain combinations of these are particularly favorable energetically. So this is typically encoded in what's called a Ramachandran plot. So this is a Ramachandran plot here, which is showing for one amino acid the distribution of phi and psi angles, in this case, accumulated across analysis of all the amino acids in a given database. We're just plotting points for positions of phi and psi. So each point would be a single phi paired with its psi angle. And what you observe if you do this is that most amino acids fall into a small number of regions in this Ramachandran plot. One of these regions, shown here, labeled with an alpha, corresponds to a secondary structure called a right-handed alpha helix. So this is one of the major energetically stable ways of folding a protein backbone. There's another region here that corresponds to beta strands. We'll see that these come in 2 slightly different variants, parallel and anti-parallel. You get some left-handed alpha helix that's not as common. That's this other region here. And of course, you get some amino acids that are pushed out of these favorable regions, but often a large fraction of a protein will fall into one of these combinations of regions. So you get a lot of these energetically favorable states that make up large fractions of most proteins. Any questions about that? So there are a couple of exceptions to this. So 2 of the amino acids are special and don't use the standard Ramachandran plot. So is anyone aware of what those are and why? Yeah.
Proline, because it has the weird ring structure that restricts it, and glycine be the other one, just the hydrogen in it.
Yeah, so proline and glycine don't quite use the standard Ramachandran plot. So if we go back here, proline has a side chain that links back to the backbone, so it has more restricted movement. It effectively occupies a smaller part of the Ramachandran plot. Glycine, the side chain is just a hydrogen, so you're able to rotate it into a larger number of positions without getting steric hindrance, that is, clashes between that side chain and other parts of the protein. So glycine occupies a larger portion of the Ramachandran plot. But basically, this is telling us what's allowable in these proteins. And that gives rise primarily to 2 kinds of secondary structure. So this is a few different images of a piece of alpha helical structure. What we see here is simply the backbone. You can see that there is this characteristic twist, the helical structure, 3.6 residues per twist of the structure. And this is just a top view of that. If we take a slightly more realistic view of this sequence, we're adding back more of the atoms. This is showing what's called a wireframe diagram of that same piece of alpha helix. So this is putting in all of the non-hydrogen atoms. And it's worth knowing what the color code is when you look at these pictures. The standard code is that carbons are represented as gray, reds are oxygen, and blue are nitrogen. So you should be able to see when you look at something like this that you're seeing a peptide backbone wrapping around. And the side chains will then project around the alpha helix. So they're projecting out around this and giving some shape to this, which is apparent in this space filling model, which is capturing approximately the van der Waal radii of the atoms. So this is favorable in part because it avoids the steric clashes that can make it for an unstable structure, and in part because this particular kind of structure allows for some hydrogen bonding between the backbone atoms here. So in particular, you can see very often you'll have a nitrogen and an oxygen positioned close to one another. These carboxyl oxygens form stable hydrogen bonds with the hydrogens connected to the amino nitrogens. And then you get these hydrogen bonds wrapping around the alpha helix, which makes it a very stable structure. So that's why we see a lot of those. Any questions about that? Right, so beta strands, as I mentioned, come in a couple of different variants. You don't usually see a single beta strand. You usually see groups of them in what are called beta sheets, and that's also driven by hydrogen bonding. And that can come in a couple of different formats. In either case, you get this very elongated backbone structure, and in some cases you'll get what's called a parallel beta sheet, where you have multiple beta strands all running in the same direction. So let's say all running from amino to carboxyl when they look left to right. That would give you a parallel beta sheet, or they could run in opposite directions with successive strands, in which case you get anti-parallel beta sheets. And you can mix those together in a single beta sheet. Again, we get a lot of these close pairings of the oxygens in the carboxyl groups with the nitrogens in the amino groups, and that gives you this favorable pattern of hydrogen bonding that makes this also a very stable structure. These will tend to have a characteristic pattern of the side chains that we can see in the wireframe diagram, where alternating side chains on any strand will point in opposite directions relative to the sheet. So one strand will be pointing out of the sheet, the next side chain will be pointing in, the next one pointing out, and so forth. And that sometimes leads to patterns in the behavior of the amino acids or the preferred amino acids that you can use to identify these computationally. Just as in the alpha helical case, when we look at a space-filling model, you see something that is a lot harder to recognize as beta strands or beta sheets, but is more realistically what you would actually see if you could look at a protein. So the actual thing is a much more irregular kind of structure than maybe the idealized model of what the secondary structure would be. Any questions about this? Okay. It's worth noting that when we look at structures, very often we'll need to look at different levels of abstraction, and sometimes we'll use what's called a cartoon view, which uses a kind of visual code to represent an abstract version of a protein, typically representing alpha helices and beta strands through these kinds of cartoon models. You can very quickly see the overall shape of a protein and see where the secondary structure elements are. For alpha helices, sometimes you see this kind of coiled ribbon to represent an alpha helix. Sometimes it's simply a cylinder, so just be aware that there are different ways of representing that. And beta sheets are often represented as these arrows pointing amino to carboxyl across the beta sheet. So in this case, we see a parallel beta sheet with two strands. It's worth noting that those are not the only kinds of secondary structures. There are some more exotic ones you'll sometimes see. So this is a collagen triple helix, a kind of more elongated version of helix that's found specific to a protein type called collagen or related proteins. There's another kind of helix you get that's called a polyproline helix, and it's called that because if you put lots of prolines in a row in a protein, they form very favorably this particular kind of elongated helix structure. And you see some proteins that have exactly that structure, as well as some more exotic ones I'm not going to go into that occur more rarely or in more unusual environments. So some kinds are found usually within membrane-bound regions of proteins and so forth. But for the most part, alpha helix and beta sheet account for what we refer to as secondary structure. So any questions about that? All right, so that brings us then to supersecondary structure. So there are a few supersecondary structures it's worth being aware of. One example of this is what's called a coiled coil, which is a kind of collection of alpha helices. So in a coiled coil, such as the one shown here, we have three long alpha helices wrapping around one another to form a higher level coiled structure. So we have the helices of the alpha helices themselves, and they're wrapped around to form a higher level helix. This is a kind of structure you often see in oligomerization domains, so domains where maybe two different polypeptide chains are each donating an alpha helix to produce this kind of pattern, and these can occur with multiple different numbers of alpha helices. You can get 2 helix coiled coils, 3 helix 4 helix. I'm not sure if they go larger than that, but a few different variants. A more exotic one is the beta helix, shown here. So this is a pattern of secondary structure where we're building a large helix where the rungs of the helix are made of beta strands. This is a much larger structure than an alpha helix, so each of these would be comparable in size to a big piece of alpha helix. This tends to occur in proteins that need to form more or less a large flat surface, for example, for recognizing a complicated molecular structure. So that's something you don't see as common as coiled coils, but is another example of a supersecondary structure. Any questions about that? Right, so that then brings us to tertiary structure. And the tertiary structure, as I said, would be the full three-dimensional fold of a single polypeptide chain. So this is a cartoon model of a tertiary structure. And can anyone tell me what's shown in magenta in this picture? What are the magenta regions of this protein?
Alpha helices.
Yeah, so we can see that these are alpha helices. We have that cartoon version of the coiled ribbon here. So we're not seeing the individual amino acids. We're seeing this at a high zoomed out level of resolution. And in orange here, you can make out the arrows that correspond to beta strands within this protein. One thing worth noting is that, again, the thing you see when you look at the real three-dimensional structure of a protein is a much more irregular, less perfect object than our maybe idealized models of what these look like. So when you see a piece of data sheet here, you can see there's a significant amount of variability in the individual beta strands. These are much different than the idealized model of what a beta sheet looks like as a perfectly flat surface. And it's always worth knowing that our abstractions are not quite the same thing as the real protein. That basically is the tertiary structure. It's also worth noting that there are pieces of this that are not secondary structure. So some proteins will have a very large fraction of them not in secondary structure, some just a little. We would often categorize that into turn regions, which are these kinds of short pieces that just get you from one secondary structure element to another, and loop regions, which are these longer pieces. So here we're seeing a long piece of backbone, probably corresponding to many amino acids, that is it's extending beyond the secondary structure, probably for some function that's just not easily evolved through these secondary structure elements. Any questions about that? All right, so that then brings us to quaternary structure. This is an example of a protein quaternary structure. In this case, 14 different polypeptide chains that form a large complex that would then be the functional version of this particular set of proteins. And again, it's important to remember that in real proteins, the quaternary structure is usually the functional version of the protein. Most proteins function as parts of complexes with other proteins, maybe other kinds of molecules, nucleic acids, small molecules, and so forth. And it may be that a given protein is actually involved at different times in multiple quaternary structures. So just be aware that if you're interested in the functional protein, it's probably something like this, and you probably need to know about how it's interacting with its binding partners in these structures if you really want to understand functionally how that protein works. Questions about that? Right, so that basically is the structure hierarchy. But there's another way of viewing protein structures that is more or less tangential to the structure hierarchy that's often more useful for thinking about things from a computational perspective. And I want to spend a little while talking about that. And that's known as a protein domain structure. So domains are small conserved regions of protein, typically conserved shapes that occur in many different proteins, often for similar sorts of functions. So an example might be a dimerization domain. So this might be a domain that has an alpha dimerization domain to allow this protein to form into a quaternary structure. You can have, let's say, a hormone binding domain or other kinds of binding domains that recognize some kind of interaction partner, form a pocket for that to sit in. And you can have various kinds of other functional or interaction domains. For example, a DNA binding domain illustrated here, where we've got a couple of alpha helices that sit nicely on the backbone of a DNA strand and would be involved in interacting with that DNA. So many proteins will have multiple different domains within them, and it's often very useful to think of a protein as a collection of these domains. So these would be a few examples. We might have one protein that consists of these domains joined by various linker regions. And there are a lot of reasons why we care about protein domains. One of them you'll get into on Wednesday when we look at some of the computational methods behind working with protein structures, and that is that it's often much easier to recognize domains than it is to recognize whole proteins. So even if we're looking at a protein that is not in the whole homologous to anything we've seen before, there's a very good chance that protein has domains that look like domains we've seen before. And there are good ways of recognizing that, so informatics methods that you'll learn about, to identify that you're looking at a domain that looks like a domain already in some domain library. And so that can give us a lot of information about a new protein we haven't had a chance to really study in depth experimentally yet. That can often give you hints about protein function. So of course, by recognizing the domains, you often can make a good educated guess about what those pieces of protein are doing, and that can often lead to at least reasonable hypotheses about what the protein as a whole is doing. So if we observed a protein that had, let's say, one of those hormone binding domains and one of the DNA binding domains I showed on the previous slide, you might conjecture that this is a protein that is involved somehow in DNA regulation, maybe transcriptional regulation, whose function is modulated by presence or absence of a hormone. So seeing those two domains doesn't necessarily tell you with certainty what the protein does, but it's a good source of hypotheses. It's also worth mentioning protein domains because they're useful for studying protein evolution on different sorts of timescales than other kinds of information we've examined. So evolution is a theme that comes up again and again in computational biology studies because a lot of computational biology comes down to the notion of comparative genomics, that we can figure out what some pieces of sequence or structure are doing based on their similarity to other evolutionarily related pieces of protein and structure. And so it's often very important to have good models of how sequences have evolved from common ancestors. We've already seen some evidence that we can use to put together these models, so to build phylogenetic trees. So we saw nucleotide sequences, for example, and amino acid sequences. And those, as we saw last week, tend to evolve at somewhat different rates. Nucleotide sequences tend to evolve relatively quickly because you can change typically a number of bases in a nucleotide sequence without appreciably changing the function. So that allows them to evolve quickly. Amino acid sequences evolve more slowly. It's generally harder to make a change in an amino acid sequence without altering the function. And protein structures would give another kind of evidence that evolves even more slowly. So the structure of a protein will tend to evolve more slowly than its amino acid sequence. And so those can give us evidence about patterns of evolution at very different timescales. Domain structure gives another sort of evidence like that, where the pattern of domains in given proteins is something you can use to track evolution over time, and that's generally going to evolve even more slowly than the structures of the individual domains. So it's not used as often as phylogenetic evidence, but it is a line of evidence available to us for looking at long time scales. It's also worth pointing out that there is a theory of evolutionary innovation, so how new functions can evolve in proteins, that depends on this notion of protein domains. And essentially the idea is that once we recognize that proteins tend to have these well-conserved domains, particular functions to those domains, you can often come up with hypotheses for how new protein functions have evolved based on swapping of these domains among proteins. So if we observe a protein that has a DNA binding domain and a hormone binding domain, we might hypothesize that maybe in the past there was one kind of protein that had a DNA binding domain and another protein that had a hormone binding domain, and some kind of genomic rearrangement happened to put these together into a single protein and then maybe create a new kind of functional protein. So a transcriptional regulator that is regulated by activity or presence of a hormone. And that could allow the organism to perform new kinds of functions and be a way of driving significant evolutionary innovation. So So that is another reason why we sometimes care about protein domains. Any questions about any of that? Okay, so that basically goes through what I think we need to know for the purposes of this class about protein structure. But I also want to talk a bit, as I said, about the issue of how we find structures. So where do protein structures come from? If we have a new protein, how would we figure out what its structure is? And there are a number of different technologies for doing this. And I want to go through a few of the major ones, because as I mentioned, when we get into looking at these, it's often very important to know what kind of structure we're looking at, because that will tell us some important things about the characteristics of that particular structural data. We would often assume that the gold standard, so the best kind of structure to have for a protein, is what's called an X-ray crystallographic structure. So X-ray crystallography begins with a crystal. So you need to take your protein and get it to crystallize, which simply means getting a regular lattice of many copies of that protein in one or a small number of different shapes. So packing repeatedly to form a crystal structure in which you have the same shape or a small number of shapes repeated over and over at regular distances. And it's worth noting that getting a protein to crystallize is not generally an easy thing to do. So there is a lot of effort that typically goes into crystallizing a protein. They don't normally naturally crystallize. Today there is a lot of automation behind this. So there are crystallization robots or robotic systems that can try lots of different solution and precipitation conditions in parallel, do a lot of the work that used to be done by hand. There's still a lot of guesswork, a lot of expert knowledge that's needed, a lot of tricks of the trade, often to get a protein to crystallize. This can be months or years of work for a particular protein, and often that will end in failure. So someone can spend years trying to crystallize a protein and get nothing out of the effort. Very often, getting it to a crystallizable form involves modifying the protein. This is something we need to pay attention to when looking at crystal structures, that what's in the database is very often not exactly the functional form of the protein or the naturally occurring native form. It's quite common that you need to truncate the protein. So if it has an end that kind of hangs off and is floppy in solution, that may interfere with crystallization, and a crystallographer may chop that out of the protein to get it to crystallize. There may be long loops inside the protein, and they'll cut those out and put in something shorter that's less flexible. You may, in some cases, need to change the sequence in various other ways, make amino acid substitutions to get it to fold, to get it to pack correctly in these crystals. And very often, it will be sensitive to ligands, so binding partners of the proteins. Proteins usually have binding partners, and sometimes a protein will only crystallize in the presence of its natural binding partner. Sometimes it will only crystallize if it's not in the presence of its natural binding partner. Sometimes it can only be brought to crystallize in the presence of some non-natural ligand, so something it doesn't usually associate with. And these are all things we have to pay attention to when looking at the structure. It may be bound to its normal binding partner. It may have multiple normal binding partners. It may be bound to some of them. It may be bound to something that it doesn't usually encounter in nature. At the end of all of this process, we may get a structure, and even if we get the structure, there is no guarantee it's actually in exactly the same shape as it takes on in its normal function in the cell. A crystal is a very non-natural environment for a protein, and that can induce changes in the structure that can be hard to predict. So these are all things to be aware of when we look at crystal structures. Any questions about that? All right, but eventually we would hope you would get to a crystal structure, and when you do, that's where the X-ray part of X-ray crystallography comes in. You shine an X-ray beam through your crystal, and what you get out of this is a spot pattern that essentially is capturing where there are irregularities in the structure. Everywhere you have reproducibly the same atom at the same distance, so some structural regularity because of this repeated packing in the crystal, you can identify an interference spot. And the goal of crystallography is to take these interference patterns and then solve the inverse problem of going back to the electron density of the original protein. There's a lot of complicated mathematics behind this. I'm not going to go into how that works here. It's pretty involved and complex. But for our purposes, it's worth being aware that this is pretty much a solvable problem right now. The computational mathematics behind it is not trivial, but if you can get to a good quality crystal, you're pretty sure that you're going to get a crystal structure out of this. You can solve that mathematics, and you can kind of thread the protein through this electron density and get a good quality structure from a good quality crystal. So again, for our purposes, that's more or less the level I want you to understand. Getting a crystal is hard. Solving for it once you've got the crystal is more or less a solved problem. And this technology has some appreciable advantages and disadvantages over the other technologies we'll be seeing. The main advantage of crystallography is that it tends to give very high quality structures. And we generally measure the quality of a structure in terms of the uncertainty in the positions of the atoms to be assigned to that structure. And that's usually measured in distances of angstroms. So if we look at what we would say is a 3 angstrom structure, roughly speaking, that means that we've placed the atoms to within about 3 angstroms of their correct positions in that structure. So crystallography is the most reliable method for getting high-quality structures. So about 3 angstroms is where we would generally say we've got a really nice structure, a really good atomic detail structure. Crystallography, in some cases, can get appreciably better than that. In rare cases, under an angstrom, which is really a fantastic structure. But it's generally going to do appreciably better than any of the alternatives we'll be seeing. And that's the main reason people really like crystallography. But as we saw, it has some significant disadvantages. It's slow, expensive, and unpredictable. What we get is very likely to be the structure of a modified version of the protein that may or may not be reliable for what the naturally occurring version looks like. And although I didn't mention it, it's especially hard to do for some classes of proteins. So generally, it's going to be hard to, let's say, get a membrane-bound protein to go into a crystal structure. Some membrane-bound proteins are very underrepresented in our structure databases. And in general, the structure databases are very biased with respect to crystal structures towards things that are easy to crystallize. Any questions about X-ray crystallography before we move on? Okay. The next major alternative people would generally return to is nuclear magnetic resonance, or NMR. So again, NMR, like X-ray crystallography, has actually quite a lot of interesting math and computing behind it that I'm not going to go into because it's really too involved and too advanced for what we can get to, I think, in this class. But I'll just say at a very high level, what this does, more or less, is give you information about which atoms are close together in a protein structure. So there are different variants of this, but a typical example would be telling you about which of the amino nitrogens are close together in a particular protein structure. And what you get out is a spot pattern like this, where each of these spots is telling you about one of these distance constraints, telling you that there are 2 atoms that happen to be close together in your protein structure. And by looking for particular patterns of spots, you can identify, let's say, signatures of particular amino acids. You can figure out from that which spots correspond to which residues in your polypeptide chain, and then figure out which of those residues are close together, which atoms in those are close together, and put together a good model of the overall 3D shape of your protein. So that tends to be another very good way of getting protein structures experimentally. And it has some appreciable advantages relative to X-ray crystallography. One of them is that this works on proteins in solution, which generally means that it's going to be, in some sense, more similar to the naturally occurring protein. When it works, it's much easier to get it to work. So if you can get your protein in solution, which most proteins normally are, then you've got a pretty good chance of being able to get an NMR structure. But it does have some disadvantages. One disadvantage is that it generally gives you lower resolution than X-ray structures. So generally, if you want to know the atomic detail structure of a protein, people would prefer an X-ray structure to an NMR structure. And it has limits on how large of a structure you can look at. There have been some big technological advances the past few years, so this keeps getting pushed further and further, what it can handle. But traditionally, it couldn't handle more than on the order of 100 or maybe now a few hundred amino acids. There are tricks you can use sometimes to get to larger proteins. But generally, if you have a large protein and you need an experimental structure, crystallography is the only alternative. It often gives you rather than one structure, a family of structures, and that is typically a big advantage of NMR. So in an NMR structure, because you have the proteins in solution, they will often have some dynamics to them. So proteins often don't have a single rigid shape, they're somewhat flexible. They may have hinge regions that can occupy, let's say, a couple of different positions stably, so 2 or more discrete positions, and they often have disordered regions that are flexible in solution, and NMR gives you a way of identifying when you're looking at these situations. So seeing that there may be some pieces of structure that are very conserved across the different conformations of the protein in solution, some that look wholly disordered, and some that may have a more restricted kind of structure, maybe allowing for a few different possibilities. And that's something X-ray crystallography typically won't give you, at least without a lot of work NMR typically will give you. So NMR at a high level has the advantages of getting you solution state structures, which is usually what you want, and giving you dynamics, which is often very interesting, but it has the disadvantage of being limited to small proteins and generally being a medium resolution in contrast to the high resolution of X-ray crystallography technology. Any questions about this? There's one more experimental technology I want to mention, although it's kind of an emerging technology, but it's really come to the awareness of most biologists in the past few years, and that is 3D structure from electron microscopy. Often very specifically what's called cryo-electron microscopy, which means electron microscopy of frozen samples. So EM has long been a technique you can use to look at static two-dimensional images. So if you look at a textbook and you're seeing an image of a cell, let's say, there's a pretty good chance you're looking at an EM image. It's less well known that for many years now, this has actually been also a technology you can use to get three-dimensional structures. And the way this works is more or less the way you or I might infer the three-dimensional shape of an object by looking at lots of two-dimensional photographs of that object. Essentially, if you can see the same object from many different angles, you can put together a model of what its full three-dimensional shape might look like. And that's basically the idea behind cryo-electron microscopy structures. For a long time, this was really just a low-dimensional structure determination method. It was something typically used for looking at rough structures of large complexes of proteins. So the virology field has used this for a long time to figure out the structures of viruses, which are fairly large objects compared to a protein, typically hundreds or thousands of proteins in them. And this would give you structures in the range of maybe 20 or 30 angstrom structures. That's very poor resolution. But the technology has really dramatically improved in the past few years. So really in the past 5 years, this has emerged as something that most structural biologists are aware of. It's still primarily something people are using to look at large complexes, so things like a virus or, let's say, a big molecular machine like a ribosome. But when you have something that's too big for NMR and that won't crystallize easily, this gives you a way of getting a structure for that. So the advances in this have come in part from advances in the experimental technologies behind the cryo-EM to get clearer images, in part just making it more high throughput, so it's now possible to get many more images. And actually a lot of the advance behind this is computational, coming up with better ways of taking large numbers of images and figuring out how to put them together to build a 3D model. But basically in the past few years, people have started to get out atomic resolution models from this. So models of things that won't be pretty close to that. So you won't see yet a lot of these in the databases, but it's getting there, and I think it's a technique you should all be aware of. Right, so that is the last of the experimental techniques I wanted to tell you about. But this is a computational class, so I want to tell you in the remainder of this this lecture about some computational techniques we have in getting protein structures. Are there any last questions about the experimental methods for structure determination before I get to this? Okay, so structure prediction basically refers to computational reconstruction of protein structures, and it more or less comes in two flavors. These are not entirely clearly defined, but roughly speaking, you can have de novo structures, and de novo means a structure built without prior knowledge. So typically this would mean taking an amino acid sequence you know nothing about and trying to figure from generically what you know about proteins, what structure this might take on. And then there is homology modeling. And homology modeling means to take a sequence you want to know the structure of and a known structure of a closely related sequence and use the known structure as a kind of template to build a model of the structure of your new sequence. These tend to be much faster and cheaper than anything you can do in an experimental lab, so often these are just easy things to try if you want to find a structure for a new protein. De novo structure prediction is something that people have been working on for decades, but it wasn't really until about 10 years ago that this actually started to sometimes work. It's still much more of a research method. It's not really something people rely upon, but about 10 years ago, the first methods started coming out that could really sometimes do a good job and get a high-quality structure directly from predicting a sequence. And a lot of interesting computation goes into how this is done. So what I'm illustrating here is a high-level view of how the first really successful method, called the Rosetta method, worked. And this relied on the fact that there are a couple of different ways that people commonly try to computationally predict whether you're looking at a good structure. One of these is known as an empirical method, and an empirical method says that a predicted structure is likely to be good if all the little local regions of amino acids packed against one another look like ways that amino acids pack against one another in libraries of real proteins. The alternative to that is what's called a physics-based method, which basically means you take your molecular model and you try to simulate the laws of physics that would act on it. Electrostatic forces, van der Waals interactions, hydrogen bonding, all of that kind of thing, and see if it looks energetically stable. And the Rosetta method combined versions of both of these to come up with the first really successful method for this. So at a high level, the way this worked is as follows. We would take a sequence for which we want to know the structure, and we would try to break it down into lots of little pieces of sequence, lots of little peptides. So we could notice that this has a peptide HVN within it, and you would then go to a database and say, what are all of the ways in a database of protein structures that we see HVN folding? We would also look for VNM and NMC, go in our databases and try to figure out all the ways those fold within different protein structures. And what we could do is put together these composites of all of the ways of combining all of the ways these local regions fold. And what that gives us is a way of very quickly searching a large conformational space, putting together all these different pieces in different possible ways to try lots of different plausible ways this protein might fold. And we could then use these empirical energy functions, these empirical assessments of whether you're looking at a good fold, to figure out that some of these conformations are plausible and some are not plausible. So you might go through a huge number of these 100,000 or 1 million different possible conformations, and then very quickly narrow it down based on these kind of low-resolution models to, let's say, 50 or so that look like potentially good structures. You then switch to a physics-based model. So you map all of the individual atoms into this, switch from this low-resolution backbone level model to a real atomic resolution model, put in a detailed physics-based force field, and do what's called an optimization. So allow this to adjust to the most energetically stable positioning of the backbone, the side chains, and all of that. And then at the end, come up with an identification of which of these are able to settle into what seems like a really nice, stable protein structure. At the end of this, you might not narrow it down to one structure. You may have maybe 5 different guesses for what might be the good structure. And if one of those 5 is a really good one, you generally say that's been successful. So these technologies have been improving quite dramatically the past few years. They actually will often do a very good job, especially on smaller proteins. but they often won't do a very good job. And for larger proteins, this really isn't something you can rely on. It does have some big advantages. You can try it on any protein, and it basically doesn't cost anything to try this. At this point, you can take an amino acid sequence, you can go to a structure prediction web server, upload your sequence, and then they'll email you back some candidate structures for your protein. It's really an easy thing to try, but it doesn't really work very well. When it does work, the resolution is usually not that good. Sometimes it will hit great, even crystallographic quality structures, but not usually. And it has a very big problem that you don't know whether or not it worked. If you're trying to get a crystal structure, generally you know if you've got a good crystal or not, if it diffracts nicely, and you know if you have a nicely diffracting crystal, you're going to get a nice structure out of it. Same with NMR, you generally know whether or not it worked, more or less. But with de novo prediction, it's always going to give you an answer, and you really have no idea if the answer it gives you, or more typically, if any of the several answers it gives you is actually in any way close to the real structure. So it's something you can try, but not yet something you can rely upon. Homology modeling is an alternative to this that is often much more trusted and much more reliable. And the way homology modeling works at a high level is we would take a sequence whose structure we want to know, and we would search through databases of other sequences whose structure we know. And if we find good alignments, let's say using the BLAST method we've seen before, then we can take those sequences of known structure and bring in those structures or the overlapping regions of them as templates and do what's called the threading to try to thread the unknown sequence onto the backbone, substituting in the unknown sequences amino acids for the amino acids of the known sequence to try to get a high-level model structure of our new sequence. You can then move to a more precise level of abstraction, so do what's called loop modeling to throw in maybe extra pieces that don't correspond to anything in our known structure, throw in all the side chains, add in one of these physics models to try to exactly position all the side chains and loops and all of that, and get it down to a really good quality structure. And this is something that actually does work pretty well a good fraction of the time. It's something that's very fast, very easy to try, and if you have a good homologue, this will often give you a good quality structure, something you can really use in a laboratory setting for example, doing structure-based mutation studies. So let's say you are trying to figure out how to mutate a protein to knock out a particular binding surface or disable a particular function. If you can find a good homologue known structure and build a homology model, that actually has a pretty decent chance of working well. So this is something that is pretty widely used, but you do need a homologous protein, and generally you need it to be a pretty close homologue. It's not going to work if you have a very distant homologue. And just like with the de novo structure prediction, it does have the disadvantage of that. You don't necessarily know if it worked well. It's going to give you an answer at the end. It may or may not be the right answer. But if you have a close homologue, there's a pretty good chance it's gotten close to the right structure. So any questions about any of these methods? Okay, so that basically brings me to a summary of these different methods. And again, remember when we get into looking at these structures on Friday, it really does matter where these come from because of the different trade-offs involved. Crystallographic structures tend to be high quality but hard to get, not always reliable, but they're usually what we would think of as the gold standard for a structure. NMR is generally easier to get but only available for small proteins. It gives us some important information X-ray structures don't, especially protein dynamics, but generally not as high quality. Cryo-EM is an technology for large complexes of proteins. It's starting to become an important way people study protein structures in detail. Homology models are a good option that is often quite reliable if you can find a good homologue. And de novo models are a rapidly advancing but still not quite trustworthy technology, but a good thing to know about and always an option if all else fails to at least get some hypotheses about the structure of your protein. So any last questions about anything before we break for the day? Okay, so just a reminder that you'll have a guest lecture on Wednesday from Joel McManus, and then on Friday I'll be back to tell you about some of the actual structure databases.